Cleanroom and contamination control best practices. Cleanroom and contamination control best practices are essential for ensuring that controlled environments consistently meet the high standards required for semiconductor and pharmaceutical manufacturing, biotechnology, aerospace, and other precision-driven industries. These best practices are categorized into 4 key pillars: personnel, environment, cleaning, and monitoring. First, personnel protocols and training are critical. Staff must be properly trained in gowning procedures, hygiene, and cleanroom behavior to prevent human-sourced contamination. Second, maintaining the environment means regulating air quality through HEPA filtration, pressure differentials, and temperature and humidity controls. Third, cleaning procedures and tools must be properly selected and regularly validated to remove particulates and biological contaminants. Finally, monitoring ensures that all standards are met through routine inspections, particle counts, and microbial testing. Together, these guidelines help protect products, ensure quality, and uphold regulatory compliance within cleanroom operations.
What is a cleanroom? A cleanroom is a controlled environment designed to minimize the introduction, generation, and retention of airborne particles. Within this space, temperature, humidity, and pressure are precisely regulated to prevent contamination that could compromise sensitive processes or high-purity products. These conditions are essential in industries such as semiconductors, pharmaceuticals, biotechnology, and aerospace manufacturing. Key characteristics of cleanrooms include controlled air filtration, often using HEPA or ULPA filters to remove particulates. Temperature and humidity are carefully regulated to meet process and material requirements. Positive air pressure is maintained to keep contaminants from entering when doors open. Surfaces and materials used inside the cleanroom are specially selected to reduce particle shedding and facilitate cleaning. Cleanrooms are also classified by ISO standards, ranging from ISO 3 to ISO 9. For example, ISO 3 to 5 are used in semiconductor manufacturing, while ISO 6 to 7 are common in pharmaceutical settings. ISO 8 applies to controlled medical device areas, and ISO 9 covers general industrial manufacturing. What is a cleanroom? Cleanrooms play a vital role in industries that require contamination-free environments to protect product integrity. These include electronics manufacturing, medical devices, aerospace, pharmaceuticals, food processing, and biotechnology. Each of these fields depends on precise environmental control to ensure safety, performance, and regulatory compliance. One of the hallmarks of a cleanroom is its high-efficiency particle filtration. Cleanrooms typically utilize HEPA filters that remove at least 99.97% of airborne particles as small as 0.3 microns. Temperature is tightly controlled, often maintained within +1 degree C, to support both process stability and material compatibility. To ensure continuous compliance, cleanroom conditions are monitored around the clock, 24 hours a day, 7 days a week. By tightly managing these parameters, cleanrooms provide a controlled space where precision manufacturing and research can take place without the risk of particulate or microbial contamination. Sources of contamination. Understanding the sources of contamination is essential for implementing effective cleanroom controls. The vast majority, 80%, of contamination originates from personnel. This includes skin cells, of which 30,000 to 40,000 can be shed daily, along with hair and fibers from clothing or the body. Speaking and breathing release respiratory droplets, while walking and gesturing create movement particles that stir up contaminants. Because of these risks, personnel are considered the primary contamination source. The second largest contributor is equipment, responsible for about 15% of contamination. Moving mechanical parts can generate particles due to wear. Lubricants and oils may leak or evaporate, contributing further. Electrical discharge from static buildup can release particles, and surface erosion from tool degradation adds to contamination risks. Other minor sources include material transfers at 3% and the external environment at 2%. Though smaller, these sources must still be managed through proper material handling and facility design to ensure cleanroom integrity. Sources of contamination. Material and environmental factors also contribute to contamination risks in cleanrooms, though to a lesser extent than personnel and equipment. Materials introduce a transfer risk, particularly when packaging such as cardboard, tape, and labels are brought into the cleanroom. Raw materials like powders and chemicals can easily disperse particles if not handled properly. Tools and supplies that aren't designed for cleanroom use may shed contaminants, and even documentation, like paper or pens, can be a source if not properly controlled. Environmental sources are external and often relate to facility design or equipment failure. HVAC systems, if poorly maintained, can allow filter bypass or failure, letting contaminants circulate. Building structures such as ceilings and walls may shed particles or trap contaminants. Outdoor air can carry pollutants into the cleanroom if not filtered effectively. Finally, improper pressure differentials may lead to unfiltered air infiltration. The key insight: personnel generate 80% of contamination, making their training and compliance absolutely critical. The human factor: critical protocols for contamination prevention. Preventing contamination in semiconductor manufacturing starts with people. Strict gowning and hygiene protocols are essential to maintaining a cleanroom environment. Pre-gowning procedures require individuals to remove all jewelry and watches, secure any long hair completely, and change only in designated areas to avoid bringing in outside contaminants. The correct garment sequence must also be followed. It begins with shoe covers, then proceeds in this order: hood, mask, and finally gloves. A full-body suit must be worn with proper overlap to prevent exposure. All seals and zippers must be double-checked for secure closure. Equally important are personal hygiene standards. Employees must shower daily and wear clean, lint-free clothing. No perfumes or cosmetics are allowed. Hands must be washed thoroughly for at least 20 seconds. To maintain cleanliness, no food, drinks, gum, smoking, exposed skin, or personal items are permitted inside cleanroom zones. Personnel best practices. To maintain a contamination-free environment, personnel must follow specific behavioral protocols. Movement within clean areas should be slow and deliberate. Sudden gestures must be avoided, as they can stir particles and increase air turbulence. Always minimize airflow disruption and follow marked pathways to maintain cleanroom integrity. Communication should be limited. Speak only when necessary. When speaking is unavoidable, turn away from critical surfaces and use hand signals when possible. Never cough or sneeze in clean zones. For entry and exit, use airlocks correctly and wait for the full cycle to complete before passing through. Only one person should enter or exit at a time, and every access event must be logged for traceability. Training is essential. Initial certification includes over 8 hours of contamination theory, gowning practice, and exams. Ongoing training includes annual recertification, monthly reviews, and retraining after incidents. Proper training can reduce contamination risk by 95%, with a 30-minute max time in critical areas. Creating and maintaining optimal atmospheric conditions. Maintaining strict atmospheric control in cleanroom environments is vital to minimizing contamination. High-efficiency particulate air, or HEPA filters, achieve 99.97% efficiency at capturing particles as small as 0.3 microns. For even finer filtration, ultra-low penetration air, or ULPA filters, can trap 99.9995% of particles down to 0.12 microns. Pre-filters remove larger debris, capturing 85% of particles at 1 micron. These filters typically last 12 to 24 months, but their pressure drop must be regularly monitored to ensure airflow remains effective. Air changes occur at a high rate, between 240 and 600 times per hour in Class 100 cleanrooms. Temperature is tightly controlled within plus calc or 2 degree F, and relative humidity must remain within plus 5%. Pressure control is also critical. A cascading gradient maintains cleanliness, with the cleanroom at plus 0.05, WC, gowning at +0.03, corridor at +0.01, and outside at atmospheric pressure. 90% unidirectional airflow and a face velocity of 0.45 meters per second help direct particles away from sensitive zones. Creating and maintaining optimal atmospheric conditions. Environmental conditions in clean rooms must be tightly regulated to protect sensitive processes. Temperature is maintained between 68 to 72 degrees Celsius. This narrow range ensures process stability and user comfort. Continuous temperature monitoring, uniform distribution throughout the space, and tailoring conditions to specific process needs are all essential. Humidity control is equally critical. The target relative humidity is 45 to 55%, which supports material stability, prevents static buildup, and maintains a comfortable working environment. Heating, ventilation, and air conditioning, HVAC systems, are the backbone of cleanroom climate control. Best practices include using redundant systems to prevent failures, continuous 24/7 monitoring to detect anomalies, and scheduling regular maintenance to ensure reliable performance. Optimizing energy use without compromising control is also important, as is installing alarm systems for early detection of issues. Success depends on integration. Filtration, pressure, temperature, and humidity must work together as one seamless environmental control system. Systematic protocols for contamination elimination. A structured and consistent cleaning schedule is essential for controlling contamination in cleanroom environments. Cleaning must be continuous during operations, performed 24 hours a day, 7 days a week. This ensures that particles are removed as they are generated, maintaining real-time cleanliness. At the end of each shift, a daily cleaning session is conducted to reset the environment before the next group enters. This includes wiping down surfaces, equipment, and entry zones. Once per week, a more intensive deep clean is performed. This 7-day interval cleaning targets areas that are not covered during daily routines. Every 30 days, a full decontamination process is carried out. This monthly procedure includes disassembly and full sanitization of equipment and infrastructure. Finally, every 90 days, a quarterly validation confirms that cleaning protocols remain effective. This systematic matrix ensures that contamination is controlled consistently across all timescales, from hourly operations to quarterly inspections. Systematic protocols for contamination elimination. Contamination control is essential in semiconductor environments, and achieving it starts with using validated, approved materials and strictly followed procedures. Primary disinfectants include 70% isopropyl alcohol, known for being fast-acting, quaternary ammonium, which offers broad-spectrum antimicrobial activity, peracetic acid, valued for its sporicidal properties, and hydrogen peroxide, which can be used in vapor phase to reach difficult surfaces. These agents are selected for their effectiveness in high-sensitivity environments. Cleaning tools are equally important. Lint-free wipes and sterile mops prevent particle shedding. HEPA vacuums capture fine contaminants, while non-shedding materials ensure the area stays particle-free. The standard procedures begin with pre-cleaning. All materials are removed from the area, contamination levels are documented, and HVAC system performance is verified to ensure proper air handling. During the cleaning process, a top-to-bottom approach is used with overlapping spray patterns and a 30-second minimum contact time. Finally, verification involves visual residue inspection, environmental monitoring, and documentation to ensure compliance and cleanliness. Systematic protocols for contamination elimination. Contamination control requires a disciplined approach targeting 4 main categories: surfaces, equipment, critical zones, and validation. For surfaces, weekly deep cleaning of walls and ceilings is essential to prevent dust buildup. Floors are wet mopped daily to reduce particulate load. Work surfaces, being high-touch areas, must be disinfected after each use to avoid cross-contamination. Equipment protocols include cleaning production tools between batches to prevent carryover between product runs. Furniture is wiped down daily to control surface-borne contaminants, and HVAC grills are cleaned monthly to maintain proper airflow and limit microbial growth from dust buildup. Critical zones demand heightened attention. Airlocks are disinfected after every entry to prevent contaminants from entering clean environments. Pass-through chambers are cleaned at each transfer, and gowning areas are sanitized at least twice daily due to high personnel interaction. Validation steps ensure cleanliness through microbial testing, visual inspections, particle counting, and documentation. These steps verify compliance, confirm environmental safety, and ensure traceability. Systematic protocols for contamination elimination. To maintain cleanroom integrity, performance standards must be strictly upheld. The target efficacy for microbial reduction is set at 99.9%, reflecting the high degree of cleanliness required in semiconductor environments. For sporicidal activity, a contact time of 15 minutes is essential to ensure complete neutralization of resistant microorganisms. Additionally, a minimum dwell time of 30 seconds is required to allow disinfectants to act effectively on contaminated surfaces. Achieving 100% documentation compliance is also critical, as it ensures traceability and adherence to protocols. Key success factors drive these outcomes. Consistent methodology ensures that procedures are repeatable and dependable. Using proper chemical concentrations guarantees disinfectant effectiveness without harming surfaces. Adequate contact time is vital to eliminate microbes. Thorough documentation supports audits and process reviews. Finally, regular validation testing verifies that cleaning protocols remain effective over time. Each factor contributes to a holistic contamination control program. Continuous assessment and compliance verification. Maintaining a clean and stable manufacturing environment requires real-time monitoring of critical parameters, ensuring continuous assessment and compliance verification. Particle levels are tracked around the clock, 24 hours a day, 7 days a week, for particles 0.5 microns and larger. This ensures airborne particulate contamination is detected and addressed immediately. Pressure differential monitoring is equally critical, with tolerances set at plus or minus 0.01. This tight control helps maintain directional airflow between clean zones and less controlled areas, reducing the risk of contamination migration. Humidity, measured as relative percentage, is held within plus or minus 2% to avoid material degradation or static buildup, both of which can compromise product integrity. Finally, airflow velocity is monitored with a tolerance of plus or minus 10%, ensuring adequate air exchange and contaminant removal throughout the controlled space. Together, these parameters form a comprehensive system for ensuring environmental control, product quality, and regulatory compliance through precision monitoring. Monitoring and validation. Monitoring and validation are essential to maintaining cleanroom integrity and regulatory compliance. Particle monitoring is conducted in real time, ensuring continuous surveillance when the area is occupied. At-rest testing is performed weekly to assess baseline cleanliness. Operational testing occurs monthly to verify conditions during active use, while full qualification is conducted annually to confirm the space meets all required standards. These tests follow ISO 14644-1 guidelines, using defined sample points and a minimum sample time of 1 minute. Microbial testing complements particle monitoring by detecting biological contaminants. Active air sampling uses volumetric samplers to measure airborne microorganisms. Passive air sampling employs settle plates exposed for 4 hours. Surfaces are tested using contact plates and swabs, while personnel are assessed through finger dabs and glove juice techniques. Acceptance criteria vary by cleanroom class. JOCR 1 CFU for Class 5, JOCR 5 CFU for Class 6, and 50 CFU for Class 7. These thresholds help ensure a sterile and compliant environment. Monitoring and validation. Monitoring and validation systems are essential for ensuring consistent quality, compliance, and traceability in controlled environments. In the data system segment, SCADA integration allows for seamless supervisory control and data acquisition, making it possible to manage complex operations efficiently. Real-time trending enables proactive tracking of environmental or process parameters to identify deviations early. Automated alerts are triggered when conditions approach or exceed specified thresholds. Historical archiving ensures long-term data storage for analysis and compliance. Additionally, audit trails in accordance with 21 CFR Part 11 provide a secure, traceable record of system activities and changes. The alert system categorizes responses into 3 levels. A warning is issued at 80% of the action limit, prompting early awareness. If the action limit is crossed, an investigation is required. A critical status calls for an immediate response to prevent risk or failure. Standards include ISO 14644 for cleanroom classification, USP for sterile compounding, EU GMP grades, and FDA 21 CFR regulations for pharmaceutical manufacturing. Validation protocols and success metrics. Validation protocols ensure equipment and systems perform as intended with 4 key stages. Installation qualification, or IQ, verifies that all components are properly installed. Operational qualification, or OQ, confirms the system operates within defined parameters. Performance qualification, or PQ, ensures the system performs consistently under actual production conditions. Continued qualification, or CQ, supports ongoing compliance over time. These stages are backed by essential documentation, including calibration certificates, test protocols and reports, deviation investigations, and trend analysis reports. These documents support traceability, regulatory compliance, and internal quality audits. Success metrics drive accountability and performance. A 99.5% minimum target for data capture ensures comprehensive monitoring. Calibration status should be maintained at 100% for accuracy. Critical alerts require a response time of less than 2 hours. The annual out-of-spec rate should remain below 5%. Effective monitoring relies on redundant systems, preventive maintenance, and continuous improvement driven by trend analysis and data review. Compliance and standards. Compliance and standards serve as the backbone for regulated environments, ensuring that all processes meet legal, safety, and quality expectations across the globe. The global regulatory framework encompasses major regional and international systems. In the United States, FDA regulations are detailed under 21 CFR Parts 211 and 820. Part 211 governs current good manufacturing practices for finished pharmaceuticals, while Part 820 focuses on quality system regulations for medical devices. In the European Union, EU GMP guidelines define good manufacturing practices for pharmaceuticals, categorized into cleanroom grades A, B, C, and D. Internationally, the ISO 14644 series standardizes cleanroom classification and airborne particulate control. These guidelines ensure harmonization across borders. Pharmacopeal standards, such as USP 797 and USP 800, guide sterile and hazardous drug compounding, respectively. Together, these frameworks ensure consistency, safety, and product quality in environments where precision and compliance are mission-critical. Compliance and standards. Compliance in regulated industries is supported by well-defined standards and codes that ensure safety, quality, and operational consistency. ISO 14644 is a cornerstone for cleanroom environments. Part 1 covers classification based on airborne particle levels. Part 2 provides specifications for monitoring compliance. Part 3 details metrological and testing methods to verify performance. Part 7 focuses on separative devices like clean air hoods and isolators, which help maintain controlled conditions. The FDA Code of Federal Regulations includes several parts relevant to manufacturing. Part 2.1 covers drug CGMP, setting standards for production quality. Part 820 defines quality system regulations for medical devices. Part 11 establishes requirements for electronic records and signatures to ensure digital data integrity. Part 600 addresses biologics, laying out standards for biological products. Industry codes also play a critical role. ICH guidelines support international harmonization. ISP baseline guides cover commissioning and qualification. ASHRAE standards ensure proper HVAC design. IEST practices define environmental testing methods. Compliance process and required documentation. A structured compliance process is essential for maintaining product integrity, safety, and regulatory alignment. It begins with risk assessment, where critical control points are identified to mitigate potential hazards. The next phase is design qualification, ensuring that the proposed design meets user and functional requirements. Installation and operational qualification, or IQOQ, involves testing equipment installation and operational parameters to verify proper functionality. Performance qualification, or PQ, confirms the system performs reliably under real-world conditions and includes continued monitoring. Finally, ongoing compliance requires regular audits, reviews, and maintenance activities to ensure long-term adherence to standards. Required documentation supports each stage of compliance. Design documents include user requirement specifications, functional specs, and piping and instrumentation diagrams. Validation protocols consist of IQ, OQ, PQ protocols, test execution reports, and deviation investigations. Operating procedures document standard workflows, cleaning procedures, and training records. Quality records track monitoring data, calibration certificates, and change control history to verify consistent system performance. Compliance and standards. Maintaining high compliance and meeting global standards is essential in regulated industries. Internal audits are performed quarterly as part of a self-assessment routine, ensuring ongoing process adherence. FDA inspections occur every 2 to 3 years, depending on risk assessments, with higher-risk sites reviewed more frequently. Annual customer audits focus on supplier qualification and product assurance, ensuring customer-specific requirements are consistently met. Recertification happens every 3 years to maintain compliance with ISO and FDA standards, confirming that systems and documentation remain aligned with evolving regulations. Compliance excellence is tracked using key indicators. A 100% protocol execution rate confirms all procedures are followed without exception. Corrective and preventive actions, or CAPA, are resolved in under 24 hours. 0 critical findings signal no major compliance failures. Data integrity is nearly perfect at 99.8%. Full training compliance is achieved, and document deviation rates stay below 5%, ensuring robust operational quality. Key takeaways and summary. Achieving cleanroom excellence depends on mastering the 5 core pillars: people, environment, cleaning, monitoring, and compliance. First, properly trained personnel who follow protocols help reduce contamination. Second, maintaining optimal air quality through strict environmental controls prevents particle intrusion. Third, regular and validated cleaning routines are critical to sustaining cleanliness levels. Fourth, continuous monitoring validates that systems remain in control. Finally, adherence to compliance standards ensures that all practices meet regulatory and industry expectations. Critical practices include recognizing that personnel are the leading source of contamination. 80% of particles originate from people. Proper gowning is essential, and HEPA filters must be maintained on schedule. Documentation must be complete. If it isn't recorded, it didn't happen. Quick wins include limiting cleanroom personnel, enforcing entry and exit protocols, offering regular training refreshers, and using signage to reinforce behaviors. High-priority investments include real-time monitoring and training, while medium-level priorities include automation and backup systems.